Hello. Here we're going to be talking specifically about the mitochondrial electron transport chain. So during the catabolism of carbohydrates, fatty acids, and amino acids, many molecules of NADH and FADH2 are produced. These electron carriers must be recycled through oxidation reactions so that catabolism can continue. This is because the oxidized form of NAD+ and FAD are required substrates for catabolism. In the absence of oxygen, fermentation will occur to recycle these. However, if possible, these high-energy electrons will be delivered to oxygen through the electron transport chain. So the electron transport chain utilizes many different electron carriers. One carrier is ubiquinone, or coenzyme Q. Its pathway is seen in the middle here within the black box. Q is able to carry two high-energy electrons. It has an intermediate, which is a semi-radical quinone, which has accepted one electron. Whenever it's fully reduced, ubiquinone becomes quinol and has two electrons. These molecules are extremely hydrophobic, which we can see by the long hydrocarbon tail. This allows both Q and QH2 to exist in the inner mitochondrial membrane. Another example of electron carriers are metals like iron and copper, which serve important roles in moving electrons throughout the proteins. Another example is the cytochromes. Now these are proteins which contain heme prosthetic groups. Heme prosthetic groups are iron-containing cyclic molecules which each have a characteristic absorbance. The iron is coordinated by 4 nitrogen molecules, usually. There are 3 different classes of cytochromes, A, B, and C type. Another important prosthetic group are the iron-sulfur complexes, or also known as iron-sulfur clusters. There are 8 different ones within the mitochondria. These are usually irons coordinated by the sulfurs of cysteines. All of these different electron carriers help move electrons through the protein complexes of the electron transport chain, or between them. These all move the electrons stepwise down the energy gradient. All the reactions of the electron transport chain are spontaneous. The free energy of each redox reaction can be calculated using the Nernst equation. We can do this by multiplying the number of electrons which are transferred, N, by the Faraday's constant, F, and by the change in the electric potential of each reaction, delta E. The electric potential for each part of a half reaction are most often pre-calculated, and the values should be in your textbook. So let's put this equation to practice and calculate the free energy for the half reactions of NADH to NAD+, and O2 to H2O, which is the net reaction of the electron transport chain. So the change in standard reduction potential, or delta E, is the acceptor E value minus the donor E value. So in this case, oxygen minus NADH. So we then will multiply that number by the number of electrons transferred, which is 2, and by the Faraday's constant, and also by -1. After multiplying these together, we get -220 kilojoules per mole of NADH. This is the energy which is used to pump protons across the mitochondrial membrane. So the electron transport chain moves electrons down its electric potential stepwise to get maximum energy. Complexes 1, 3, and 4 pump protons from the mitochondrial matrix into the inner membrane space across the inner mitochondrial membrane. NADH will feed directly into complex 1, and going from complex 1 to 3 to 4 gives a total of 10 protons pumped per molecule of NADH oxidized. FADH2 is fed into complex 2, which delivers electrons directly to Q without pumping protons. Therefore, for each molecule of FADH2 oxidized, 6 protons are pumped across the inner mitochondrial membrane, as complex 2 doesn't pump any protons, so there is 4 pumped from complex 3 and 2 from complex 4. Moving protons against their gradient requires energy. So to establish this concentration gradient of protons, the energy of the electrons is used to pump protons against their concentration gradient. So the energy is transferred from one form to another. It obeys the first law of thermodynamics. So here's an overview of the mitochondrial electron transport chain. Complex 1 will oxidize NADH into NAD+. Complex 2 also oxidizes a succinate into fumarate. Both complexes 1 and 2 will reduce ubiquinone to quinol. Quinol will travel through the inner mitochondrial membrane to complex 3, where it's oxidized. Complex 3 will reduce cytochrome C, and when cytochrome C is reduced, it will travel along the membrane to complex 4, where it's oxidized. Complex 4 will reduce the terminal electron acceptor, oxygen, to create water. Now let's look closer at each step of this pathway. So the nice thing about all of these complexes is that the name clearly states what the net reaction is. So NADH ubiquinone oxidoreductase will oxidize NADH and reduce ubiquinone. So the first step of oxidizing NADH occurs with a redox reaction with FMN, also known as flavin mononucleotide. So every time something is oxidized, that means that something else has to be reduced. FMNH2, the reduced form, has 2 high-energy electrons, which will then give the electrons to the iron-sulfur clusters. The iron-sulfur clusters can only hold 1 electron at a time, so the movement of electrons will occur twice per NADH with 2 single electron transfers. Ubiquinone is able to hold 2 electrons, as we saw earlier, where it has a semiquinone radical intermediate in between the first and second reduction reaction. Within complex 1, there are a total of 4 protons pumped from the N, or negative side, to the P, or positive side. The hydrogens are moved in a specific direction, which is called vectoral movement. So complex 2 is succinate ubiquinone oxidoreductase, which oxidizes succinate and reduces ubiquinone. Now, this is actually the enzyme that is part of the citric acid cycle, but it can also gain electrons with fatty acid oxidation. The electrons will travel around 40 angstroms through the iron-sulfur clusters to get to a quinone. This mechanism is similar to complex 1, but instead of FMN, it uses FAD, and this complex does not pump any protons. Ubiquinone cytochrome C oxidoreductase oxidizes quinol to ubiquinone and reduces cytochrome C. So quinol, or QH2, will deliver electrons to the cytochrome B inside complex 3. A series of redox reactions will transfer the electrons from cytochrome B to the RISC protein, which contains an iron-sulfur cluster, and then to cytochrome C1, which is still part of complex 3, and then to cytochrome C containing protein. The cytochrome C containing protein, shown as Cyte C, is a free cellular protein which is located on the inner membrane space of the mitochondria. It associates with the inner mitochondrial membrane and moves from complex 3 to 4 once it's reduced. Once again, this is referred to as vectoral movement. Now, cytochrome C containing protein can only hold 1 electron at a time, so for each NADH molecule or for each succinate molecule oxidized, the reduction of cytochrome C will happen twice. So cytochrome C will then travel to complex 4, cytochrome oxidase. Cytochrome oxidase will oxidize cytochrome C and reduces oxygen. So cytochrome C carries 1 electron to this complex, and in this complex, the oxygen molecule is considered the terminal electron acceptor for the entire mitochondrial electron transport chain. So initially, the cytochrome C will deliver the electron to the copper A center. The copper will then reduce cytochrome A, which will then deliver the electron to the heme A3 and copper B center. The A3 copper B center will deliver electrons to molecular oxygen one at a time. So there is a radical oxygen intermediate, which is usually kept tightly bound, but as some will escape as radical oxygen species, which damage cellular components. So for each pair of electrons delivered to complex 4, 2 protons are pumped. Also, to help maintain the proton gradient, 2 protons are consumed on the negative side, or the N side, in the production of water. This doesn't add to the protons on the outside, or the P side, however, it does help keep the gradient larger. So, now that the electron carriers have been oxidized, and water has been reduced via the electron transport chain, there now exists a very large chemical gradient across the membrane. There is, on the P side, a large concentration of protons, and on the N side, a very low concentration of protons. ATP synthase will utilize this gradient and the movement from high to low concentration of protons to create ATP molecules through proton motive force and oxidative level phosphorylation. If any point of the electron transport chain is inhibited, all of the molecules ahead of the stopping point will be oxidized as the electrons will continue to flow to oxygen, the terminal electron acceptor. Anything behind the stopping point will remain reduced since the electrons will back up kind of like a traffic jam. So a good practice of working through the electron transport chain is actually to imagine breaking each point, figuring out what would stay oxidized, what would become reduced, and any other cellular consequences such as inhibition of other metabolic pathways, or if ATP synthase would still be able to continue, if the electron transport chain would still be able to continue, or if anything else would happen. It's just good practice to help you get familiar with each step and trying to work out why each step is important. Thank you.